Deep within the brain, a cluster of neurons in the lateral hypothalamus operates as a motivational activation engine. When these neurons fire, they coordinate the raw biological imperatives required for survival. In routine clinical practice, the hormones produced by these neurons, the orexins, are frequently classified as a binary switch that controls sleep and wakefulness. This diagram outlines the actual physiological footprint of the orexin system. Radiating outward from the central hub, the network exerts direct regulatory control over feeding behavior, nociception, and cardiovascular responses. As this evolutionary history details, the G-protein-coupled receptor family for orexin traces back to Placozoa and Cnidaria, some of the earliest multicellular organisms on Earth. The system operates as a polysystemic integrator. It reads current metabolic states, senses environmental stressors, and translates that data into immediate, whole-body action. Understanding orexin requires looking past the pillow. It provides the biological foundation for global physiological tone. The entire signaling network originates from a single precursor peptide, pre-pro-orexin. Looking at this sequence map, pre-pro-orexin is cleaved into two distinct peptides, orexin A in blue and orexin B in pink. These peptides possess distinct geometries. The orexin A chain is tightly coiled by two disulfide bonds, contrasting with the linear structure of orexin B on the right. To exert their effects, these peptides bind to two specific G-protein-coupled receptors embedded in target cell membranes. This schematic highlights their asymmetry. Orexin receptor 1 shows strict binding affinity for orexin A. Orexin receptor 2 is non-selective, accepting both peptides equally. Once bound, they trigger different intracellular signaling cascades. OX1R primarily couples to the GQ pathway to drive cellular activation, while OX2R couples to both GQ and GIGO pathways, introducing complex inhibitory functions depending on the target tissue. Because of its disulfide bonds, orexin A is highly resistant to degradation, allowing it to distribute widely and maintain measurable concentrations throughout human cerebrospinal fluid. A single precursor creates two peptides that interact with two asymmetric receptors. This mechanical divergence provides the mechanism a single class of molecule needs to govern vastly different physiological outcomes. The production of these peptides is highly centralized. The neurons that synthesize orexin are packed densely into the lateral hypothalamus and the adjacent perifornical area. This sagittal brain schematic maps the extensive efferent projections radiating from those hypothalamic neurons. The pathways demonstrate direct wiring into the amygdala, the hippocampus, the medulla, and outward across the central nervous system. This distribution map identifies the specific receptor endpoints. The colored markers indicate dense populations of OX1R and OX2R in critical regulatory centers, like the locus ceruleus, the raphe nuclei, and the ventral tegmental area. By terminating here, orexin directly dictates high-level behaviors, flooding the ventral tegmental area to drive reward-seeking, or the locus ceruleus to force immediate arousal. The system's reach extends well beyond the brain. Here we see the descending pathway, where orexin signals travel from the paraventricular nucleus down through the spinal cord, directly modulating sympathetic output to the heart. Further down the endocrine chain, this schematic traces orexin A as it stimulates the HPA axis, acting directly on the adrenal cortex to trigger the release of cortisol during stress responses. To calculate these outputs, the central hub requires continuous incoming data. This box shows humoral factors like circulating ghrelin, glucose, and leptin, feeding metabolic status back into the orexin neurons. Orexin acts as the master physical bridge, taking raw neurological states and translating them into immediate cardiovascular and endocrine output. When this network fails, the clinical presentation depends entirely on which part of the projection map is compromised. The most obvious failure point is the regulation of sleep pressure. These graphs illustrate the pathophysiology of narcolepsy. On the left, normal orexin maintains a stable boundary between wakefulness and sleep. On the right, orexin deficiency causes that boundary to collapse, resulting in a fragmented intrusion of sleep states into waking hours. Conversely, chronic hyperactivation of these exact same pathways, specifically the projections into the ventral tegmental area, drives the relentless motivational activation characterized by chemical addiction. In Parkinson's disease, The network failure is multifaceted. This infographic highlights how the loss of orexin impacts not just sleep, but degrades critical neuroprotection and central motor control. As the neurodegeneration of Parkinson's progresses into later stages, we see a measurable decline of orexin levels in both cerebrospinal fluid and blood plasma. Systemic failures also compromise basic survival reflexes. This schematic details the cardiorespiratory homeostatic reflex pathway, where orexin acts as the central processor for CO2 and oxygen sensing. From that processing hub, orexin regulates respiratory rate by modulating the phrenic motor neurons that physically drive the diaphragm. Sleep attacks and movement disorders and respiratory suppression present clinically as entirely separate problems. In reality, they are predictable results of a single regulatory system losing its grip on arousal and autonomic homeostasis. Beneath these systemic behaviors lies a deeper layer of control. Orexin dictates the internal survival mechanics of individual cells. This flowchart maps the bridge between orexin and cellular metabolism, the PI3K, AKT, mTOR pathway. Orexin binding directly modulates mTOR. Following the tumor branch, orexin A binding to OX1R drives programmed cell death, apoptosis, in malignancies like colorectal and prostate cancers. This OXR1 summary box reveals a complex paradox. Depending on the specific cellular environment, OX1R activation can either promote cell proliferation or trigger apoptosis and reduce tumor volume. Looking at the metabolic branch, the orexin-mTOR connection directly governs pancreatic beta cell function, determining the volume and efficiency of insulin secretion in the presence of glucose. Through these same intracellular channels, orexin A is able to suppress cytokine production, exerting powerful anti-inflammatory effects across damaged tissue. This complete pathway map visualizes the density of the network. Orexin receptor activation triggers hundreds of cascading protein interactions, gene transcriptions, and metabolic shifts simultaneously. Orexin signaling governs the internal survival mechanics of individual cells, directly coding metabolic and survival responses at the tissue level. The current pharmacological tools we use to manipulate this system are dual orexin receptor antagonists, or DORAs. This class includes suvorexant, daridorexant, and the available medication, lemborexant. Clinically, DORAs offer a highly favorable safety profile. They present nil addiction liability and carry a lower risk of patient falls compared to traditional benzodiazepines. Yet, a clinical paradox exists. Despite the vast systemic reach of the orexin network, DORAs are prescribed almost exclusively for the treatment of insomnia. Based on the anatomical projections and receptor affinities we've examined, utilizing DORAs merely to induce sleep leaves the majority of their therapeutic capacity untapped. Because orexin heavily modulates the basal ganglia and central motor pathways, DORA medications present a strong disorders and complex tardive syndromes. This illustration models the nociceptive pathways, demonstrating how targeted orexin A modulation along the descending spinal tracts can directly intercept pain signals triggered by ischemic stress. Furthermore, by dampening the hyperarousal circuits in the locus coeruleus, DORAs are uniquely positioned to treat severe clinical anxiety and paroxysmal sympathetic hyperactivity syndrome. This ability to globally downregulate autonomic storms makes the mechanism effective for stabilizing the erratic neurochemistry found in post-head injury patients. This system controls the body's primary motivational and autonomic tone. The neuroanatomy clearly demonstrates that orexin modulators function as high-impact tools for treating complex systemic and neurological diseases.